Hi everyone, welcome back to Nanome. Today I want to talk about some of the LSD derivatives that are on the novel psychoactives market. And specifically, what the derivatives that we'll be looking at have alkyl substitutions in this position right here on the protonated nitrogen. So let's look at how some of these derivatives have different chemical structures, and then we'll look at how they bind to serotonin to a receptor differently from one another. So this is, of course, LSD. Let's pull up one of the analogs called ETH-LAD. So this over here is one of the LSD analogs or derivatives called ETH-LAD. And the difference between ETH-LAD and LSD is basically, if I want to superimpose the structures like this, we can notice that ETH-LAD is different from LSD in the position right here. LSD just has a methyl group, whereas ETH-LAD has an ethyl group. So we'll see how the difference in this having a 2-carbon chain versus a 1-carbon chain docks differently to serotonin to a receptor. The next LSD analog that we'll look at in this video is Proad, and instead of having an ethyl group in that position, it's going to have a propyl group. And let me get this in the right orientation to overlay it so that we can see how the propyl group sits differently than a methyl group. And so we notice in this position right here, overlaid in yellow, is Proad. So 1 carbon, 2 carbon, 3 carbons for a propyl group, and thus the compound Proad. So the compound ETH-LAD is different from LSD in that instead of just having a methyl group coming off of that protonated nitrogen, we have an allyl group. So 1 carbon, 2 carbon, 3 carbons, but the 2 carbon to the 3 carbon is not an alkane, it's an alkene, which makes it slightly different from the compound Proad, but also very similar. So now that we looked at the differences in the structures of these lysergamides, let's see how they dock to serotonin to a receptor differently from one another. So we've looked at this G-protein coupled receptor in a few of my other videos. This is the crystal structure of the serotonin 2A receptor, but this was created by a homology model versus the actual PDB-ID, and that's because a lot of the PDB-IDs have fake regions of protein that are used to stabilize the structure, but I kind of wanted to put more some of the real regions in there to make it more of a complete receptor. So let's shrink this down just a little bit more. Okay, and let's put LSD inside the binding site. Okay, so this is LSD inside of the binding site, and as you would imagine, there should be a hydrogen bond between these two atoms. But anyways, the this is representative of how LSD docks to serotonin to a receptor that we've talked about in other of my videos. And, there's a hydrogen bond between the indole hydrogen and the oxygen right here. And there's also a salt bridge between this positively charged nitrogen and one of the negatively charged oxygens to create a nice strong interaction there. And in fact, that's one of the strongest driving factors that forces LSD to have an interaction with the aspartic acid here. It's about a 17 kilocal per mole stabilizing force, so it's probably one of the anchors that first interacts when LSD binds to serotonin to a receptor. So that's LSD. Let's overlay that with some of the other docked poses. Okay, so if we look inside the binding site, we have ETH-LAD overlaid with LSD. The ETH-LAD is in green, and what we notice is quite interesting. And basically what happens is, there's some hydrophobic amino acids right here, which is tryptophan, and right here, which is phenylalanine. and the extra carbon, being just, CH3, hydrophobically interacts with both the phenylalanine and the tryptophan to form hydrophobic interactions, and it kind of buries itself deep into this pocket, which is a nice stabilizing force. Okay, Proad. Okay, great. So if we can see, we have now we have Proad overlaid with LSD, and Proad is in yellow. And what we can see is the same sort of story, is that the hydrocarbon chain sort of buries itself deeper into this hydrophobic pocket consisting of tryptophan and phenylalanine to hydrophobically interact with those amino acids. So we can kind of gather that the longer the hydrocarbon chain we make at this protonated nitrogen position, it just buries itself deeper into this pocket to stabilize itself inside of the serotonin to a receptor. And because there's not much of a difference between Proad and ETH-LAD, we may as well look at ETH-LAD. Okay, so we've got Proad overlaid with ETH-LAD, and what's really cool to notice is that because these compounds are very similar in structure, Proad just has a propyl group, and ETH-LAD has an allyl group, which just means that the compound in blue really has an alkene from this carbon to this carbon, which just essentially, if you know a little bit of organic chemistry, an alkene is going to have a double bond, and thereby it's going to have a stronger bond between those two atoms, and thus the bond length will be slightly shorter. Okay, so what's really would be really fun to do an experiment on is, I'd be curious to hear about people that have actually used both Proad and ETH-LAD, and kind of if there was a cool quantitative way of measuring the subjective differences between these two psychedelics. I suppose the one way you could do it in research is you could give both these compounds to animals, and you can measure the head twitch response, and you could see if the potency of one of these compounds is greater than one another. I'd imagine they probably have very similar potencies and very similar efficacies, being that the only difference is going from an alkene to an alkane. I think that'd be something really interesting to try and measure inside of an animal model, as well as hearing about people's experiences with both of these different psychedelics. And, the reason that these compounds are so interesting to me is that because inside of the body, pharmacokinetically, when you put an alkyl chain at this position, these compounds don't break down into LSD. They get inside of the brain as is, and they bind to the receptor in this, basically this exact way. So there's really not many pharmacological differences, but a lot of papers have shown that if you substitute ALT52, where you have sort of a ketone in this position, these compounds, when you put hydrocarbon chains here, these do get enzymatically cleaved and turned back into LSD. So in my perspective, these compounds aren't as interesting as ETH-LAD, ETH-LAD, and Proad. Okay, since somebody in the last video asked me to show a surface in virtual reality, let's look at the surface. For me, the main reason to look at a surface of a receptor versus, just looking at it in its alpha helical and beta sheets structure is that you can actually see with the surface how the ligand gets inside of the active site of the receptor. And if we spin this around a little bit, we'll probably see some holes inside of the receptor, usually in the extracellular space that the ligand can snake its way or wiggle its way in there. Let's give a look here. So here's one. So here's one very small hole right here. This is there is a hole likely that LSD does not get through here because it would have to, I think its rigid benzene rings would have its rigid cyclohexane / benzene rings would have a really hard time squeezing its way in here. But look at the top of the receptor. Yeah. So this is a much more likely candidate for how LSD gets inside the binding site or orthosteric site of the receptor. There's quite a large hole in the top of the receptor near the extracellular space where LSD could wiggle its way in and actually get inside of the active site. Maybe for fun, we'll just go inside the active site just for a second. Not as interesting. Thought that might be cooler. Let's turn this off. Actually, this is pretty interesting. Let's pull this up a little bit. If we look inside of the active site of the LSD molecule, we can kind of see the surfaces that LSD's lysergamide backbone are interacting with. If we look kind of at the wall of the receptor in gray, and we can also kind of see how the diethylamide group right here hydrophobically interacts with surfaces near itself too. You know, with that being said, with that being said, I hope you guys enjoyed that video, and I usually look for the comments to see what people want to see next in some of my videos. Usually can kind of come up with ideas as I'm doing PhD work. You know, ideas kind of just come to me when I'm doing molecular docking within Nanome. And, you know, I hope you guys have had the opportunity to actually use Nanome inside of virtual reality. I think the price of an Oculus headset just went down to $250 in the US, making them more accessible to buy. So I really hope VR takes off in education, because as you guys know, I'm very bullish, so to speak, on the fact that VR hopefully will be used in education soon, because, you know, I wish when I was taking organic chemistry in college, this was available, because it really has changed how much easier it is to learn organic chemistry and understand how molecules sit in three-dimensional space. Till next time, stay curious.